So first, hi guys, how are you? um, so what did I want to say? So from the tutorials, there were some questions that came up. First one is, if I'm using the 11th edition of the book or 10th or whatever, can I still work with that? So for the theory, you can work with an older version, okay? Because in the span of 10 years, so since 2012, they went from 10th to 12th, okay? So there's not much changes. The problem is when I'm going to assign to you practice problems, the numbers will be different between version 11 and 12. Okay, so what I suggest you do is go to the library, the course reserve of the library, get the 12th edition, and then just make sure that you have the right numbers. Okay. Now in the tutorial, you don't really need to bring the textbook, the physical textbook, as long as you have access to the problems, be it on your computer, tablet, phone, whatever, you can do that. Okay. The second question that came up from the tutorials, at least the first two, is the quizzes. So myself and the other and the TA, we were asked if the quizzes will be similar to the questions that were assigned for the tutorial or not. They will be similar in terms of, so for example, for those that did like question 38 and 39, the short answer will be in that style, okay? For the multiple choice, they will be similar to what you've done in the tutorial, but they will be based on the whole lecture, okay? Not just like I'm gonna give you a problem in the tutorial and then the exact same with different letters in the tutorial. It doesn't make sense, right? So that's pretty much it for the questions. They're not gonna be as hard as 65 or 67, okay? Those are more like exam questions and even then, okay? But still, study for it. What else? What else did I want to tell you? The lectures. Okay, I finally succeeded in putting the lectures online. Well, at least the first one. So now it's going to be much more smoother to put the others. Okay. Did anyone see the whole thing? You got it? Okay. So just go to the link and then I'll put the lectures. Hopefully, the next day. Depends, because it takes like 10 hours to do its stuff, right? So I'll let it run while I'm working, and then whenever it's done, I'll upload it. Okay. Makes sense? Okay, so next week you'll start the quizzes for the tutorials. So you'll be with your real TAs. For this week it's a bit special, but next week you'll be with your TA. Okay? You guys have any questions? Yeah.
Are there specific stuff on the Achieve platform that are required?
No. The Achieve platform, the reason why I'm using the Achieve platform is because the book is very expensive. Okay, that's why there's people using the 11th edition, 10th edition, used, old, okay? Like, I don't want to make you spend like $350 just for a book that you're gonna use one semester, okay? So the Achieve gives you the textbook and gives you more questions to practice instead of you going on Google and looking for question banks or, Course Hero or whatever, okay? So that's the only reason why I'm using the Achieve platform, but it's not mandatory, okay? The quizzes, on there, the adaptive quizzes are important for me because when I go on there and I see that half the class failed, like the questions related to 3.3 of the textbook, then I know that in my next lecture I need to focus on that, okay? And it shows me which questions you failed, so it tells me what topic you're kind of weaker in, okay? Those ones are also harder, so if you're getting like, because I've seen people do them already, if you're getting like 65, 70%, you're already doing pretty good because it's covering a lot from the book, okay? You're not expected to ace those on the Achieve platform, okay? Make sense? Questions? Yeah? Okay, so let me tell you, actually, we'll do a vote now, okay? So the midterm, I know that you have one with Samantha on the 22nd, right? So my two options are to put it on the Tuesday before that, which is 3 days before, so the 19th or 18th. Anyways, the Tuesday of that week. Or I can put it 1 week after the break. No? You don't want to study in the break? So you know what? Let's do a vote. Who wants after the break? you guys got beat. Lift up your hand properly so I can see. Okay, who wants Tuesday? Okay, deal. Tuesday it is. Okay, I'll change that on Moodle. Okay. Yeah, that's better because there were some profs that like to put work during the break, and I used to be annoyed by it, so I understand. So we'll do it before, and then give you a free week. Okay. Any questions related to the course? No. Okay. So today we're gonna learn about the second law of Mendel. But before I do that, I'm gonna just revisit some concepts for single gene inheritance, so that we can then it's gonna be easier for us for the next part of the lecture. So last week we were talking about dominance and recessiveness. We have to be a bit more specific about that. So the alleles can be changed, right? If you're saying that one is dominant, the other is recessive, and then the way you write it can change, right? And so when we're talking about recessiveness, what we mean is that the mutated allele is the recessive allele, so the small m in this case, okay? And this happens in haplosufficient genes. So if you have a heterozygote in a haplosufficient gene, that means that one allele is sufficient to make a healthy phenotype, okay? So then the mutated allele would be considered the recessive because you would need two of it to make the mutant phenotype. In a haploinsufficient gene, so a gene that requires both healthy alleles or wild-type alleles to kind of give the healthy phenotype, in that case, the mutated allele is the dominant one. Because whenever you have a mutation, just one mutation in the allele will be a dominant phenotype because you won't have enough copies of wild-type gene. Does that make sense? Okay. So then when we write the conventions on like your exams or whatever, there's different ways to do this. Either you can use a capital letter, or you can use, for the wild type, so you can use capital letter, or you can use a plus sign, okay? Or you can write the letter and then a superscript, which is a plus sign, okay? So these things, you'll see them in the questions that will be assigned to you, and it's important that you kind of know that a plus sign means wild type, and then a small letter means mutant, okay? Makes sense? Good. So I'm gonna just like go not in depth, but a bit about the molecular. Let me just put this down here. I'm just gonna talk a little bit about the molecular idea behind alleles and mutations. Okay. I think you all did like CGEP bio, or at least yeah, you probably did that. So you understand that genes are just a sequence of DNA, like DNA molecules. Okay. So a mutation in the gene could be just a deletion of part of that gene, of that sequence, or a substitution or whatever, okay? So then different alleles have different sequences of DNA, right? And then depending on where that mutation happens, then it will have a certain specific effect. For example, if you look on the right here, I'll just do it like this. If you look on the right here, if the mutation happens in an exon, which is the part of the gene that codes for protein, it might mess up the protein or make it better, right? It's not always just mess it up. Sometimes when it gets into the active site, it could make it bind the substrate much better, for example, okay? And then different mutations lead to different mutant alleles, okay? So that's pretty much it. We'll get back into this into a lot more in depth. I think it's chapter 7 or so, I don't remember, but we'll talk about this more in depth, okay? So our little buddy Mendel, okay? He created the first law, and now there's a second law, the law of independent assortment. And this law, what it says about genes is that genes, so obviously we don't have only one gene, we have 20,000 genes. So these genes, they move between parent to gametes to offspring in a specific way, right? So genes that are on different chromosomes, they sort independently, okay? They're not dependent on each other. But genes that are on the same chromosome, if they're very close to each other, they will be inherited in the same, like together, okay? And this is chapter, I think, 4 that we're going to do this, okay? One little exception to this is the ends of chromosomes. So the ends of chromosomes are super far apart, so they're considered kind of different chromosomes. So genes that are apart from each other on the ends of chromosomes, sometimes they are inherited independently. Okay, and we'll see these three scenarios in the next lectures. So this guy decided to study genes, how they are transmitted to the offspring together. Okay, not just a single one and then focus on that single one. Okay, we'll get into his experiments, but I just want to give you a little bit of the cool part of the lecture, let's say. Okay, so Mendel's study on how genes are inherited together led to the creation of genome-wide association studies, or GWAS. Okay, and this is just a way that we use in genomics to kind of examine how genes associate together. Okay, and then they define a trait. And you can pretty much go online if like you sit down and look for it. There's the whole genome that's sequenced for humans, and then you can find the same with a diseased person, for example, and then compare the differences between them. And then you will see why this one is healthy and this one is diseased, right? So one interesting thing that these studies found is that people with blonde hair have an increased susceptibility to skin cancer, okay? Which you would not find because they're two different traits, like unrelated things, right? But there is a relationship in the DNA that we can't figure out just by studying like peas or whatever, right? You need some huge database of DNA that you can compare with, let's say, different people, okay? I don't want to scare people who have blonde hair. It's a lot more complex than just this. But if you're interested, there's a paper here that links hair color with skin cancer, and then using the Mendelian genetics, okay? Cool. And there's a lot of interesting links like this. You can go online and find them. You know, it looks weird, right? Blonde hair, skin cancer, why? What's the problem there? But they still don't know, but they know that there's a link there, okay? What we can do also with GWAS is find specific mutations in genes, and then these mutations will tell you, for example, if a protein is much better or weaker, right? Things like this. I'll talk about GWAS a lot because there's a lot of interesting things to do with this, and it's kind of the way we work right now. Okay, back to Mendel. So our buddy Mendel studied pea plant shape and color as the two genes to kind of study how they assorted together. And yeah, before I get into the details. Okay, so when we're talking about two genes or multiple genes, there's a way that we write it. So the convention that I was talking earlier, if we're talking about two genes that are on different chromosomes, we have to separate them by a semicolon. Okay, remember that we are diploids. So if we have a heterozygous like this, that's gene A, and then they're on different chromosomes, so we put semicolon and then heterozygous for B. Okay, if they are on the same chromosome, we remove the punctuation and instead we put the heterozygote together. So AB is one chromosome, and then the diploid is the other one. Okay. Now, genes that are on the same chromosome, they don't get inherited the same way as the ones that are separate chromosomes. Okay. So this is like a place where people make mistakes during exams because they don't focus on where the gene is. Is it next to the other, or are they on different chromosomes, or what's happening? If we don't know, then we will put a dot between them. Okay. People like to trick students with this. You don't know, and then you have to figure out if it's linked or unlinked and all that. Okay. We'll see how to do that in the future. Okay. Now that I read the hybrid cross, one of the students asked me last lecture, I think, what's the difference between monohybrid and heterozygous, and I was a bit freaking out, so I didn't answer it properly. But monohybrid refers just to the cross, okay, and heterozygous represents the individual, so the two copies of the gene. Okay, I don't know who asked me, but that's the answer. Okay, so when we're talking about dihybrid, we're talking about a cross between two genes, two genes, and then if it's trihybrid, it's 3 genes, etc. Okay. So when we get into the how do you call it? When we get into the transfer of genes from one parent to the offspring, we have to always figure out the gametes. That's the most important part. Okay, step number one has to always be for you when you're solving these problems, find the gametes. Okay, so in that case, we have, let's say we have a double heterozygote that will produce gametes, so then you need to make all the combinations of possible gametes. Okay, I left some white space here, bought a pen for this screen, apparently it doesn't work, so we're going to do it on the board, and I have to figure this out another time. So I'll just show you what I mean. So when we are doing a cross, is it this one? Okay, what? Okay. So when we're doing a, so if we have, for example, two genes, A and B, okay? And let's say they're on different chromosomes, okay? They're on different chromosomes. If we're looking to do a cross with these guys, the first thing that we need to figure out is the gametes. Okay, so here they're talking about a heterozygous, so we can just do this. So now, how many combinations of gametes can you have? 3, 6, close guys. Okay, so we have big A, and then two options, right? We have the B, and then the B, right? So then it becomes AB, and then AB. Does this make sense? Right? And then same thing for the small a, right? So A, B, and then let's see, A. So there's 4 different gametes. Does that make sense? Okay. So this is how you figure out your how do you call them? Your gametes, okay? Before you do the cross. And if there's a third gene in there, then you need to include it. So then it's going to be like this, right? And whatever. Make sense? If let's say we have C or D, and then you just keep going like that, and then it becomes huge like this, okay? You won't have this like super huge like this in the exam, but there will be questions with like 5 genes, okay? Just not the whole cross because it's a waste of time, okay? One thing that's important is that the 4 gametes that are produced here, they will occur at equal frequency. That's rule number 1, okay? The law number 1 of Mendel. And then law number 2 is the independent assortment, which is this one can assort with this or that, this one with this or that. Make sense? Okay, let's go back to this. Okay, so 1 second. So what Mendel did is he took his piece and then he looked at 2 genes, okay, the shape of the seed and the color of the seed. And the first thing that he did, he mated them together. So he took 2 pure lines. Now, when we're talking about pure lines in the dihybrid, we're still talking about homozygous, either dominant or recessive, okay? There's no heterozygote in the parental line, okay? Now, if you look at the top here, actually, let's look at it like this. So if you look at the top, from the green one, the round green, you can only produce one type of gamete, right? Big R, small y, okay? And the big R gives you the round shape, small y gives you the green color, and then big Y gives you the yellow color. Okay, so he crossed it with another pure line, which was wrinkled and yellow, and these are the gametes produced. Hopefully that's clear. And this gave a heterozygous individual. Okay, does this make sense up to now? Okay, cool. So now he then crossed or self, no. Yeah, self. So he self this one. Look at self, I guess, and it gave these numbers. Okay, so when you do this for a dihybrid cross, what happens is that you'll end up with these ratios of phenotypes. Okay, so he found that there's 9/16 round yellow, 3/16 round green, and then 3/16 wrinkled yellow, and then the most recessive one is the wrinkled green. He found only one of them. Okay. If you look at these numbers, this is just a 3-to-1 ratio inside a 3-to-1 ratio. Okay, and I'll show you why. So if you get a problem like this on the exam, this is what exactly you have to do. Okay, you'll take the 2 parental lines, you'll make you'll figure out the gametes, and then from there you will separate the gametes in a table, and then you can just go through them one by one. So the first thing you'd say, like the top left square of the table would be homozygous dominant for the R and homozygous dominant for the Y, and then from there you can infer the phenotype, right? Does this make sense? So you would do this, and then you can do it for all of them, okay? And you get this right here. So I think this is clear now. Makes sense. Easy stuff. Okay. Cool. So that's basically it. And then you'll probably be asked to figure out the percentage of the offsprings or the probability that you'll get a certain genotype or phenotype. Okay. One thing that the TA told me is that you had a little bit of difficulties with the, your ability to predict the progeny ratios. So there's two rules to follow. Okay. There's the product rule. The product rule means that if you're looking at two events, okay, event A, event B, and you want them both to happen. So both to happen, you have to multiply the probability of one to happen by the other, okay? If I take a coin and I want two heads to come one after the other, well, the probability that one head comes out is half, 1/2. And then for it to happen again is half, so half times half equals 1/4. Does that make sense? Okay. The sum rule is when you want two events that are exclusive to happen, so either or. So what is the probability that I get either heads or tail? So it's half for the heads, half for tail. Add them together gives you 1, right? You'll get either heads or tail. Does that make sense? Okay, this is the same thing. So if you're doing a question about this, you'll just try to find the keyword or and, okay? Either or and, okay? And these two will tell you if it's a product rule or if it's a sum rule. People who are in computer science, you'll use this a lot. Yeah. Yeah, and is product, sum is sum. Sum is or, I mean. Yeah, makes sense? Good. Okay, that was the easy stuff. Now let's get into the serious stuff. So now if we are asked to predict progeny ratios, for example, in this question, it says, what is the probability of obtaining a fully homozygous recessive for 5 genes from a cross with 5 different genes with another individual that has 5 different genes? So questions like this look complicated to answer. Let me move this thing here. I think we'll use it. Okay. So the way you solve this question, because we're talking about independent assortment, you can treat them separately. So you start with the A, so the heterozygous A, and then, so let me actually do it on the board. 1 second. While we're at it. What? Just 1 sec. Okay. Okay, so it's asking you what's the probability that you get a fully, actually I'll write it, okay. So we want this guy. This guy, so B. Jeez, okay. I'm not going to write the cross. I think you have it in your notes there, but I'll tell you what they're giving. So you separate them by gene, okay? So the first thing, we want a specific genotype from 2 parents, okay? So then we need to multiply the probabilities of getting the genotype for A, the genotype for B, C, D, and E, okay? We multiply them all together, okay? So if we start by the first one, the parents are of type AA crossed with AA. Okay, so what's the probability of getting this from this? 1/4, right? If you do the Punnett square, you'll get 1/4. So this one is 1/4. Next one is small b, big B. Okay, what's the probability of getting this from these two? Half, right? If you do the Punnett square, you'll figure out that it's half. Okay, then you do it for C, D, E, and then you multiply them all together. Do you have any question about that? Easy? Okay, so just remember in the exam or in the quizzes or whatever, just take it step by step, okay? Remember that these genes sort independently, so you can treat them separately from each other and then go back to the whole question, okay? What else do I have to tell you about this? Yeah, I think that's pretty much it. What people like to ask also in, and probably me, I don't know, in questions is, what is the probability of, let's say, this happening and it being a male? So then you also multiply it by half, right? Because you can either be male or female. Okay? Yeah, that's pretty much it. Okay. Just 1 second. As you can see. Okay, a different way to ask this question. So another way we can another thing we can ask you about this is, for example, how many different genotypes will we produce? So if we're looking at a monohybrid cross, it's quite simple, right? You're able to count, let's say, for example, it's 2 heterozygous individuals, you'll make 3 genotypes, right? Homozygous dominant, recessive, and then the heterozygous. But if you have 5 genes, then it starts being a little bit more complicated, right? So if we look at the cross here, right here, actually, you know what? Let's do this one here. Let's do this one here. So AA, for example, the third one, right? So if you're crossing big A with big A, small a, how many genotypes can you produce? 2, right? Either homozygous dominant or heterozygous, right? So then you put that in the equation. So 2 multiplied by, and now if we look at all the 3 other, what do you call them? All the other 3 other genes, so B, C, and D, they're all heterozygous crossed with heterozygous. So how many genotypes can you produce from these? 3, right? So 3 to the power of 3 because there's 3 genes. And you multiply all this together to give you the number of distinct genotypes. If we look at the second one, the one right above it, you take the genes separately again. So the heterozygous for A with the heterozygous for A, homozygous recessive for A, how many genotypes can you produce with this? Only 2, right? Okay. Same thing applies to B, same thing applies to C, same thing applies to D. So then it's 2 genotypes to the power of 4 because there's 4 genes. Make sense? Easy? Okay, I have to increase the level here. We're going to be done in a few minutes like this. Okay. Okay, there's a different way of doing this, of solving this problem. These problems, okay? When you have 2 or more genes, you can do the branch tree that I just showed you. Okay, so let's say, for example, we're crossing a homozygous dominant for both genes with basically the 2 pure lines, okay, for both genes. We'll get a heterozygous for the 2 genes as offspring, and then if we self it, it can become complicated to determine like the genotypes, all the genotypes. So what you can do is take the first gene, right, cross it with itself, take all the possibilities, right, and then put them on the first column for the gene 1. So if you cross heterozygous RR, oh, this works. Can you see? Okay, put that. Okay, nope. Okay, I won't do it with my finger then. So if you cross 2 heterozygous for the R gene, so then you'll have 1/4 homozygous dominant, 1/4 homozygous recessive, and half of it is heterozygous, right? So you just put it like that, and then you figure out the second gene for, which is the same thing basically. So if you have homozygous dominant for the R gene, and then you look at the gene number 2, you'll have the same probabilities, right? 1/4 is 2 big Ys, half is heterozygous, and the other 1/4 is homozygous recessive. Okay. Now, if I ask you what's the probability that you'll get 2 homozygous dominant for the genes, then you have to multiply the probability that you will get homozygous dominant for the first gene by the probability that you'll get homozygous dominant for the second gene, which is 1/4 times 1/4, which gives 1/16. Does that make sense? Okay. Can you do this without calculator? Yeah. Go ahead. Why, when you're so when you're crossing this one here with itself, that's selfing, right? Yeah. So this is what selfing means. Make sense? Can you do this? Yeah. Wait, this one here? This is just me. I'm giving it to you like this. Okay. Well, you need the first one, right? You can't. This one here. Right, so, okay, so you make the gametes, right? So if you make the gametes, the first one can only give you a big R, big Y, and the second one can only give you a small r, small y. And if you do the cross between them, you'll end up with heterozygous. So I'll do it actually. I like using this board. Let me just load this thing. 1 second. Okay, so the first one there, so it says, so we have big R, Y like this, right? So the only gamete produced from here is RY like this, right? And then if you cross it with this guy, the only gamete is this, right? And so if you do the Punnett square, which is R, Y, you'll end up with this. R, Y, right. What am I doing? Wait 1 second, let me figure out what I'm saying. Oh, it's not a one thing Punnett square. Just 1 second, guys. I don't know what the hell I'm saying. This is bad. Okay, let me think. Yeah, sorry, I'm sleeping here. So this is the only gamete, right, that you can make, right, from this one and from this one. If you combine them together through fertilization, then you'll end up only with this here. I don't know why I was making a Punnett square, but does this make sense? Haploid, diploid. I don't know why I went to Punnett squares. I guess I like Punnett squares. I don't know. Okay, back to this. So then you self it with itself, and you'll end up with all this. Okay. If you have a third gene in there, you would do the same thing. Treat them separately and then go through the thing. Yeah. How come you self it? Okay. Well, you don't need to self it, but if we're studying inheritance of genes and we want to see how they are transmitted, then you start with the pure lines, you'll get the offsprings, and then you'll go to the second generation, third generation, right? And this is what Mendel did. This is his experiment, okay? It's not like I'm selfing it just for fun, right? This is his experiment, okay? If, for example, in the exam, I ask you to self it, this is what I mean, right? Now I'm just, maybe I was not clear. I'm presenting his experiment, okay? Cool. No. No. Is that clear, guys? You good? So everyone's going to get 100%. Okay, cool. Yeah. Wait. So this is not on the same chromosome. If they were on the same chromosome, the calculation becomes like much more complicated. Then we'll learn it, I think, not next lecture, maybe next week. Okay, you have to calculate the distance and then the probability of them being transmitted together or not. So this is linked inheritance. It's a bit more complex than this, but for now it's only the independent assortment. Okay, so this assumes that they're on separate chromosomes. Okay. Okay, so this is what I just said basically. So then if we're counting the chance that how many of them will happen that are both round and yellow, then you have to add all the probabilities. Okay, because different genotypes can make the same phenotype, so you add them up. Just like different size of a coin can make the same coin. Okay, and so you just add them, and this is what you get: the 9-3 to 3-1 ratio. Okay, good. All right, test cross. So then, if we're doing a test cross, oh, my slide is messed up here, but I guess you can understand that. So a test cross for two genes is the exact same thing. You're test crossing with a pure line, be it homozygous dominant or recessive. And then if you do the Punnett square, you start with the gametes, always with the gametes. So the right side can only give you small r, small y. The left side can give you big R, big Y, big R, small r, big Y, etc. And then you put them on each side of the Punnett square, and you do that. Okay. So if you remember from our first or second lecture, we were talking about a single gene inheritance, and it had a phenotypic ratio of 1 to 1, right? If you test cross it. While here, it's a 1 to 1 to 1 to 1. Okay. I'm not going to ask you to remember the genotypic ratio for. Sorry. Yeah. Not necessarily. You can still use the other one, right? If I do big R, yeah, you can use any of them. Okay. Now the genotypic ratio for this, not this one here, but the other one is a bit more complex. It's like 1 to 2 to 1 to 4 to whatever. Okay, don't remember it. Just be able to kind of figure it out by doing this here, and then you just count the genotypes. Okay. Make sense? I mean, there's no sense in asking you a question to tell me the genotypic ratio for like this is like useless, right? You're never going to use it again in your whole life. But yeah, if you ever get a question on the tutorials or whatever, you know what to do. Okay, so what is the segregation pattern from this cross? Now, I'm not asking you about cross. I'm asking you about segregation pattern. Okay, so can you guys tell me? How do the genes segregate? Any idea? It's just a different way of asking you to form the gametes, right? Does that make sense? Okay. So then all you have to do is do pretty much the same thing, which is big A, big B, big C, big A, small b, big C, and then go there, okay? If you want to count them, this is how you do it. So the first one on the side can produce 8 gametes. I think you can probably do it, but it's going to be big A, big B, big C, big A, small b, big C, big A, small b, small c, etc. There's 8 of them, okay? Same thing for the other ones. So if you try to figure out the gametes, you'll end up with 4. And then how many possible patterns? There's 32 in total, okay? Because big A, small a can produce how many types of gametes? Either big A goes with the genes or small a, right? And then you multiply them for the others. If it was homozygous, for example, recessive, like the second one, small b, then there's only one gamete produced from that gene, right? Does that make sense? Okay. Good. More branch stuff. Okay. When you have problems, try to solve it like this. And then if you, so let's say we do that pattern there, you can see all the 32 different genotypes. Okay. Next, if we ask you what's the probability or what is the frequency of offspring with the genotype above? So heterozygous for the A gene, homozygous recessive for B gene, and homozygous dominant for the C gene. Do you know how to do this? Sorry? No, you can do the tree. Yeah. So what you do is you separate them again. We're asking you for a probability of this specific to happen. So that means it's the probability of the AA happening, big A, small a, multiplied by the probability of the homozygous recessive for B times the probability of the homozygous dominant for C. Okay? If you do this, you can do just that part of the branch. Okay? Of the whole branching system. So let me tell you, the first one is heterozygous for A, second one is heterozygous for A. So the probability that I get a heterozygous for A is half, right? Because you do the Punnett square, and then the diagonal is AA, right? So that's half. Then you take gene number 2, which is B. If you're crossing big B, small b with two small b's, what's the probability that you'll get the homozygous recessive? Half. So that's that right there, right? And then you do the same for C, you'll get a quarter. And then you multiply out all that, it will give you 1/16. So that's the answer for the question. Let's say, for example, I wanted you to use the sum rule instead of the product rule. I will ask you, what is the frequency that the offspring will have this genotype, or the same thing for A, the same thing for B, but for C, but B, I want it homozygous dominant. So then you'll take the probability of both genotypes, and then you'll of happening, and then you'll add them together. Does that make sense? Because now I'm asking you for the probability of having either or of the genotype. Okay? This is why gamblers fail. They think I think it's easy. You know, I had a friend that used to gamble a lot, and he thought that he was winning the game, but he doesn't understand that there's like 8 packs, and then the probability that you get this specific card. In his head, it made sense, right? But no good. Okay. Last part of the lecture. Okay, this double muscle problem. Okay, so there's this gene that makes the muscles of cattle much bigger. Okay, double muscle, right? And it's controlled by a dominant allele, DM. So if you get DM, you have more muscles. If you get the recessive allele, you'll get normal muscles. And then there's also a second gene for the coat of the cattle, okay, which is controlled by a dominant allele, big B. So if you have big B, what do you get? Dominant, right? So you get black fur. If it's small b, you get the white color. Does that make sense? So how would you solve a problem like this? How would you start by like, well, not the problem is not yet defined, but if you get this information already in the question, start writing the genotypes, like, you know, the DM means that you have the double muscle, big DM means that you have double muscle, and then small DM you have small muscles, whatever, okay? So then they ask you, the question comes like this, okay? So a white double muscled bull, homozygous for both genes, is crossed with a homozygous black, normally muscled cow, okay? So from this, are you able to figure out the genotypes of the two parents? Okay, so this is the first thing that you do. Then they say, then for you to figure out the offsprings, you kind of need to get the gametes, right? So you get the genotype of the parents, you figure out what gametes they produce, and then you go on from there, okay? And then they will start, I will ask you probably questions like, what's the probability of getting a black double-muscled individual in the F2? And so step by step, you try to figure this out, okay? I left the right side to write on it so I can, you know, do it there, but the pen is not working. So we're just gonna go to the next page, okay? This is how you solve this problem. The first line here, a white double muscle, blah, 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 blah. You will get it, you will start by figuring out the parental genotype, okay? So white double muscle is basically 2 small b's and 2 big DMs, okay? And they're saying that they're homozygous for both genes. So if the DM is dominant, that means it's big D, small m, and then homozygous, so you have the same thing for both sides. Now the other parent is the black normal muscled parent, let's say. And so black, we say it's a dominant allele also, so you have a homozygous dominant, which is big B, and then normal muscled, so small dm, okay? Don't get confused if you see, for example, genes being written in like 2 letters, 3 letters, 4 letters. It means the same thing as just writing D and B, okay? It's just depending on the organism you're working with, you'll have different ways of writing it. For example, yeast, you'll have 3 letters, etc. Okay? Does that make sense? Humans, you get to 5 letters, but the 5 letters are all the same gene. Okay? Good. So then they're telling you to cross these two. You have to make the gametes from them, right? So the gametes for the first parent is small b, big dm. And then the other one is big B, small dm. Okay? That's the only possible gametes you can produce from these 2 parents. Is that clear? Yeah? Okay. Okay. Yeah. Trying to figure out if people are following or not. Okay. And then from these gametes, you produce only one type of F1 generation, right? Which is the heterozygous for both parents, from both parents. I don't know if you see the pattern that's happening here, but you get homozygous, and then you end up with heterozygous, which is the F1, and then you self it, and then you end up with 9, 3, 3, 1. Okay? This is the same thing everywhere. There's like no different way of doing it. Okay? Okay, so now we have the F1, and you need to figure out the probability of recovering a black double-muscled individual in the F2. Then you have to do the same thing, right? Figure out the gametes from the F1. So there's all these options. And then, did it mess up my thing? Okay, it's messed up. So big B, big DM, big B, small dm. The third B there, the B there alone is supposed to be with the dm at the bottom. Okay. And then if you look at this, you have 4 different types of gametes. You self them together, you'll end up with a 9, 3, 3, 1. Something like this. Beautiful drawings, okay? The big circle there, big black circle, is basically saying that it's a black cattle with big double muscles. And then the flattened one is no muscles. And then white cattle is, well, the white ones, okay? So when you have this little table here, then you get to this question saying, what is the probability of recovering a black double-muscled individual in the F2? So what you can do is just count the black double-muscled individuals. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 over 16. Make sense? Okay. What's the probability that it's a black double-muscle male? That's it. Make sense? Good. Same thing for the second question, or question number 7. What is the probability of recovering an individual homozygous for both black color and double muscle trait? So homozygous, black color, and double muscle trait. So there's only this guy, right? That's the only homozygous for both genes, right? Okay. So 1 out of 16. Okay. I think this one I just asked you. If the probability of recovering a black double-muscled individual in the F2 is 1/16, what is the probability of recovering a black double-muscled male in the F2? So it's the probability of getting that genotype times the probability of getting a male. Okay. Wow, finished. That was way too fast. Okay, I still have to figure out my timing because this is my first time, right? So I don't know if you guys have any questions or not, but that's pretty much it for today. Next lecture, I will add more slides, maybe 60 or okay. Cool. See you on Thursday. If there's nothing. Yes.